Right? So in a dicot plant, these are the arrangement of xylem, the vascular bundle basically. This is the vascular bundle. This whole can be this is the vascular bundle. This is present in the roots. These are the vascular bundles which are organized in a certain manner in the stem, and this is the vascular bundle in the leaf. This is the vascular bundle in the leaf. All right, all of them are the vascular bundles. So this can be regarded as vascular bundle. This can be regarded as vascular bundle, and this can be regarded as vascular bundle as well. Or you can say in this picture, this is actually the vascular bundle. Vascular bundle, right? So in the root, you have to understand in the root, this is the arrangement of xylem and phloem. All right, this is the arrangement of xylem and phloem. We're talking about dicotyledonous plant. When we come to the stem, we see that in a dicot plant, the vascular bundle basically arranges itself in a form of ring. So this is the stem. You can say this is the ring where the xylem and the phloem are going to arrange themselves. All right, in this manner. All right, this region, the innermost region, is called the pith. This is the pith, and then there is uh, this is the vascular bundle that we have been talking about. Vascular bundle, vascular bundle. They are arranged in this circular manner, and we need to know that for each vascular bundle, there is xylem, and there is phloem. All right, so this is the phloem, and the inner part is the xylem vessel. Is this clear? The image is clear to you? For each of them.
जी
For each of them, this is the inner one is the xylem, and the phloems are going to be at the outside. The phloem, phloem, and phloem. Right? All together, this is called vascular bundle. The arrangement needs to be understood. But if you talk about a monocot plant, which is not really I'm just giving you an understanding. It might appear in the exam. Monocot, monocot plant. In monocot plant, the vascular bundles are arranged in randomly like this. You understand? The vascular bundles, basically, they are arranged. This is each vascular bundle, each vascular bundle. They are arranged in a random uh, distribution, right? So that is not a concern at the moment, but I just want you to know this, understand this. They might give you a question and then they might also share that with you. But this is the arrangement that you have to keep in mind. So if you look at this part of the stem, you will see that we have epidermis, which is the outermost part, the epidermis. And then we have the epidermis, the collenchyma cells. Collenchyma cells. The collenchyma cells, which are older cells. Okay, collenchyma cells are kind of older cells which have grown now and they are moving. They are transitioning from becoming the epidermis, the dead part of the outer bark. Are you able to get this, Adia? Hmm? The collenchyma cells are the ones,
जी
the second layer from the outside. Basically, these are the cells which are now going to be thickened and they are going to become the bark, the outer part. And then there is the parenchyma cells. Both together, the parenchyma and the collenchyma are going to make the cortex. All right, the cortex, the thick part, the cell that generally we see. All right, this is then there is the pit, then there is the vascular bundle, and then between the vascular bundle. So we can write a description about it a little bit. So the vascular bundle organization in stem, basically. How is the vascular bundle organized in the stem? So in a dicot stem, in a dicot, which means dicotyledonous. Basically, a dicot plant is the one, they're distinct. We are going to write the differences between a dicot and a monocot plant. Basically, dicot plants are the plants which generally they grow as two leaves when they first come out of the seed. All right. The monocot plants, they generally have one leaf like this when they come out of the the stem, the seed. You understand? So this is just one leaf, like most of the grass. Have you seen the corn or sugarcane? Those are monocot. They have leaves which are like this, long leaves like these. All right. But as for the other flowering plants like mango, like all the other plants, you're going to see that they have leaves which are like this. They have veins like these. You understand? They just have one single vein running right through between them. No branches, nothing. The leaf is like that. So this is the difference between monocot. This is the monocot. This is the dicot. This is where the word comes from. Di means two in the beginning, and mono means one, cotyledonous, which means the growth from the seed. Do you understand this? Hmm?
जी.
All right. Okay. In a dicotyledonous stem or plant stem, xylem basically, xylem and phloem are grouped and form a vascular bundle, right? So in stem, vascular bundle is arranged in a ring form. In a ring form around the pith, right? The central region pith. The phloem lies outside, xylem lies inside of the ring. Are you following me? Are you writing this down? Hmm? And in between this is the xylem outside, this is the phloem, and in between there is a band which is called cambium. This is xylem, this is phloem, and this is called cambium. Cambium. This is important. Why? Basically, cambium does what? Cambium basically, the cambium cells, they can divide and differentiate to form new xylem and phloem. All right. Cambium cells can divide and differentiate to form new xylem and phloem. All right. And these cambium cells are also responsible for thickens the stem. All right. Are you following, Adia?
जी.
So then there is the region of, so there is cambium now. cambium. Right? Together, this is all vascular bundle. Right? After this vascular bundle part, we have the cortex, right? The cortex region, which was made up of parenchyma and collenchyma cells, right? So
Hmm.
cortex and pith both serve as store serve as of food storage of food substances such as starch. All right, and then the stem is covered with epidermis and waxy cuticle layer to prevent evaporation, to reduce evaporation, you can say reduce evaporation. All right, have you written this?
G.
Some important point about roots, you have to keep in mind that when we talk about the roots, basically in the roots, this the vascular tissue, the phloem and the xylem and the phloem are not, basically they are not bundled. All right, they are not bundled, they're just there. Unlike the stem, they get bundled and more organized, but they're not bundled in the roots. Okay,
Hmm.
let me know when you're done writing. Similarly, the cortex in the xylem or in the roots also helps to do what? To store. All right. One of the important points, I'll just bring another picture. This picture is very important. See, So when the roots, we talk about roots is all covered, the root, the stem is all covered. When we talk about the leaf, if you look at it, you have to keep in mind that the xylem is always at the upper side. All right. This is something that you need to memorize. This is the xylem, right? Xylem is always at the upper side. It's always at the upper, near the upper epidermis. The one which is towards the upper epidermis is the one which is xylem. Okay, the xylem, and the phloem is the one which is at the lower epidermis, which is towards the lower epidermis, the phloem. All right, together they form vascular bundle. Basically, such drawings, this drawing is actually a plan diagram. Plan diagram only shows, only differentiates between, I've already told you that it only differentiates between the tissues, the different layers of tissues. So it differentiates between, so it is, what is it differentiating? If you look at it, if you look at it, so this layer basically is the upper epidermis, then this layer is the palisade mesophyll, and this layer is the spongy mesophyll, and this layer is the lower epidermis. You see, when we were studying this, the structure of the leaf, we also differentiated it in this such manner, right? Can you recall? I told you that needs to be differentiated in four major tissue differentiations. This can be something that might be asked from you very frequently. So please make sure that you have this in mind. This is the cross section of a leaf in which you would be required to differentiate, or you would be required to point out the xylem and the phloem. So you should be able to do that. Okay. Okay. Is this clear? Should we move ahead? Should we move ahead? Gee, can you hear me? Hello?
जी।
All right. Let's move ahead. So another thing that we have generally is the translocation. How is the translocation basically uh, the study? How can we study the path the food substance takes through a plant? All right. Basically, uh, transportation of manufactured food which are going to be what? Sugars, amino acids in a plant is called translocation. All right, translocation. The movement of food particles, the food substances, right? And we know this translocation can only take place where? Hmm? Any idea? This can only take place where? Yeah, It can only take place in phloem.
जी
Hmm? This can only take place where? In phloem. The translocation can only take place in phloem, right? Hello.
जी
All right. So that so how there are two methods to study this translocation: the movement of food substances through a plant. We have two methods. We use in one of them we use uh, aphids, aphids, and the other is isotopic method. All right, in which carbon 14 is an isotope, a radioactive isotope that is used. The movement of that isotope is then tracked and understood. The movement of the food particle that can be understood. Let's study both of them one by one. Using aphids to in translocation studies. In studying translocation. All right. So what basically aphid is a small insect. Aphid is a very minute insect, very small. Basically is an insect. It feeds on plant juices. Feeds on plant juices. It has a long mouth part which is called stylet. Has a long mouth part called stylet. All right. All right. So a mouth part. It has a mouth part which is called stylet. It's like a the stylet is like a long protruding straw or a needle which has a hole at the end, and it can take juices from there. So what this insect is actually going to do, what aphid does is that it inserts its stylet. It inserts its stylet deep into the stem until it reaches the phloem. right? So what this insect is going to do, it basically inserts its, this is the phloem, this is the xylem. So what it does, it inserts its the stylet until it reaches the phloem. All right. So what happens is, obviously, once the aphid is introduced, it is going to be feeding the. We'll write this down, inshallah. The aphid, once it has inserted its stylus into phloem, it can be anesthetized. Anesthetized with carbon dioxide while feeding. All right. The stylet, the stylet can be then cut off the fluid, the liquid will come out of the end. The analysis of this fluid or liquid shows a great amount of, shows that it contains starch and, sorry, it contains sucrose. It should be sucrose, not starch. Why sucrose? Can you answer? Because sucrose is the transportative form of, is the transportative form of the carbohydrates, and in phloem it is the translocation or transportation that is taking place. So there cannot be starch. It could be, should be uh, sucrose. Is that clear? So basically, this is one of the things. This is one of the ways to analyze that it has reached there. But the important part is that under microscope, under microscope, it it is seen that the stylet, sorry, this the stylet is inserted into phloem sieve tube. So this is in these 2 points together, They act as a proof of translocation in phloem. All right. Mhm.